Hello everyone, so today we're going to be looking at human brain size and intelligence. There's this myth that the bigger brains, the more intelligent we are. There are multiple papers and journals that do support this claim. However, with the advent of new neuroimaging technology, we are able to map out and identify various brain regions that play a role in intelligence. So how do we measure intelligence? Well, this is based on our ability to think and reason, as well as solve problems, and also on memory. There have been some correlations that have been found between brain volume and intelligence in some papers. However, brain volume seems to be one factor that has been highly exaggerated in our culture. In 2007, Young and Hea came up with the parietal frontal integration theory, and this theory sheds light on various brain regions that play a role in intelligence in humans. In their subjects, they administered the Wechsler Intelligence Scale, which is an IQ test for both adults and children. They also used these neuroimaging technology that we talked about, and found that in regions of the dorsolateral prefrontal cortex, the inferior and superior parietal lobule, the anterior cingulate and regions with even temporal and occipital lobes were correlated with intelligence in humans. One of the things they found was that variations in brain structure explain the differences that we see in intelligence among human beings. And also in 2007, a group of researchers set out to find relationship between the corpus callosum thickness and intelligence. To their subjects, they administered the Wechsler Adult Intelligence Scale and also used a Tesla scanner to observe their brain activity. They measured callosum thickness as the distance between 100 surface points from a midline to callosum top and bottom. They found correlations between total brain volume and full IQ, as well as one between total brain volume and callosum thickness. They used both of these measures to assess the relationship between callosum thickness and IQ, and found correlations between the thickness of the corpus callosum and intelligence across the posterior surface of the corpus callosum. This can be seen in both figures to the left and right. On the left, we're looking at correlations between total brain volume and callosum thickness. Structures shown here are of the corpus callosum. All the way towards the left would be the anterior end, and towards the right would be the posterior end of the corpus callosum. As can be seen on the posterior end, the purplish colors signify a more positive relationship between the total brain volume and callosum thickness. And towards the right, in the entire sample, we do see the same trend of a higher significant relationship between the callosum thickness and intelligence across the posterior end. And when the sample was separated into both males and females, we still see that same trend across the posterior surface of the corpus callosum. In 2016, researchers observed relationships between cortical convolutions and general cognitive ability in 440 adults and 662 children. The wide range achievement test was administered, as well as the Wechsler Intelligence Scale. The Jaffification Index was used to quantify brain jaffification, and this was calculated as a global whole brain measure or regionally as a local jaffification index. These were calculated of each of 198,812 nodes per hemisphere of the pile brain surface. They computed relationships between the local jaffification index and general cognitive ability in the entire sample, as well as in specific brain regions. What they found was that there were positive correlations between the local jaffification index and general cognitive ability in areas of the prefrontal cortex, the inferior parietal lobule, the temporal parietal junction, insula, fusiform gyrus, cingulate cortex, and medial temporal occipital junction. And these can be seen in the image on the left here. NIMH represents the adults and PNC represents the children. So across the test, across all the tests that were administered, we do see high brain activity in those regions that were previously mentioned. And this supports the PFIM model that was proposed by Young and Hea back in 2007. They also found negative relationship between age and the Jefferson Index, so age did not play a major role in this relationship. Finally, other factors that have been known to contribute to human cognitive ability involves the influence of hormones, such as stress hormones and sex hormones. High stress levels affects the hypothalamic pituitary adrenal axis, which leads to an increase in glucocorticoids that might reduce the performance of declarative memory by indirect damage to the hippocampus playing a role in memory. Sex hormones, including androgen, also induces neurogenesis in the hippocampus. Dopamine, also found in regions 3, 4, and 6 in the prefrontal cortex of the brain, plays a major role in motor planning, reasoning, and overall general intelligence. Synaptogenesis, our neurons' ability to establish new synapses, helps to improve the processing of information. And finally, there's also the influence of our gut microbiome. This revolves around the idea that our microbes, our gut microbes, are able to produce neurometabolites or neurochemicals, which play a role in brain function and neuronal performance. These are papers and articles that were used in making this presentation, some of which were easy to read and fun, others not so much. If you are interested, you could take a look at them.